So we begin this chapter by talking about the general features of RBCs, their shape, their size. So I'm not going to go through that, but the important thing to remember is the concentration of RBCs in males and females. So the average concentration in men is about 5.2 million per cubic millimeter, and in women it's 4.7 million per cubic millimeter. And the concentration of hemoglobin is 34 grams per 100 ml of RBCs. So this concentration does not rise above this value because this is the metabolic limit of the RBC's ability to form hemoglobin. And next comes the average concentration of hemoglobin in males and females. In males, the average is 15 grams per deciliter, and in females, it's 14 grams per deciliter. Another unique feature of RBCs is that they have excess cell membrane for the amount of material inside them. So this allows the RBC to be deformed, and because of the excess cell membrane, the RBC isn't stretched as much, and this prevents the cell from rupturing. Next comes the site of production of RBCs. So in the first few weeks of embryonic life, the RBCs are produced in the yolk sac, and these RBCs are nucleated. And then in the second trimester, the liver is the main organ for the production of RBCs. But the spleen and lymph nodes are also involved. And in the last month of gestation and after birth, the RBCs are produced only in the bone marrow. So the bone marrow of all bones produces RBCs up to 5 years of age. Then after 5 years of age, the bone marrow of the long bones starts to become filled with fat, except for the proximal humerus and tibia. And this bone marrow no longer produces RBCs after age 20. So after age 20, the RBCs are produced in the bone marrow of the membranous bones, like the vertebrae, sternum, the ribs, and the ilia. So the process of erythropoiesis in the bone marrow begins from the pluripotent hematopoietic stem cells. These pluripotent hematopoietic stem cells form different colony-forming units, which are basically committed stem cells. So in the production of RBCs, first you have the production of colony-forming unit spleen, then colony-forming unit blast, and colony-forming unit erythrocyte. After colony-forming unit erythrocyte, you have the actual process of erythropoiesis, and these different stages are PB, pore, proerythroblast, basophil erythroblast, polychromatophil erythroblast, orthochromatic erythroblast, reticulocyte, and the erythrocyte. So the different features of these in the different stages of the RBCs, you have in the proerythroblast stage, the cell has a large nucleus and 2 or more nucleoli, and the cytoplasm, which is basophilic. And in a basophil erythroblast, it's slightly smaller, and the nucleoli have disappeared, and chromatin begins to condense. Then in the polychromatophil erythroblast stage, hemoglobin begins to appear in the cytoplasm, and the cell stains with both acidic and basic dyes, and that's why it's called polychromatophil. And in the orthochromatic stage, the nucleus becomes condensed to a very small size, and eventually this nucleus disappears and its remnants are removed from the cell. Then you have the reticulocyte, which has no nucleus, but it has remnants of the Golgi apparatus, the mitochondria, and these reticulocytes eventually they leave the bone marrow and they enter the blood capillaries by the apodices. And then after 1 to 2 days, the remaining basophilic material inside these cells disappears, and after that they become transformed into mature erythrocytes. And the normal concentration of reticulocytes in the blood is less than 1%. Erythropoiesis is regulated by proteins called growth inducers and differentiation inducers. The function of the growth inducers is to stimulate the growth and reproduction of the different stem cells. An example of this growth inducer is interleukin-3. The interleukin-3 is it promotes the growth and reproduction of all the different types of committed stem cells, like colony-forming unit, spleen, blast, erythrocyte. And then you have the differentiation inducers. The function of these proteins is to stimulate the differentiation of a committed stem cell into a specific type of blood cell. And the production of growth inducers and differentiation inducers is controlled by factors that lie outside the bone marrow. For example, low oxygen in the blood would eventually cause the production of these growth inducers and differentiation inducers, which would then control erythropoiesis. So now we come to the role of erythropoietin. Erythropoietin is a very important glycoprotein hormone, and its function is to stimulate RBC production. Erythropoietin is produced in response to hypoxia, which means a low partial pressure of oxygen in the blood, and this can occur at high altitudes or when a person is suffering from cardiac failure or pulmonary diseases. Basically, any factor that decreases the partial pressure of oxygen in the blood would stimulate the production of this hormone. So as you can see, this hormone is produced in response to factors that decrease the oxygenation of blood, like a decrease in blood volume, blood loss, hemorrhage, anemia, low hemoglobin, decreased blood flow, pulmonary diseases. All these factors decrease tissue oxygenation, which would eventually lead to the production of erythropoietin. So 90% of this erythropoietin is produced in the kidneys, specifically by fibroblast-like interstitial cells surrounding the tubules in the cortex and medulla, and also by the renal epithelial cells, and the remainder is produced by the liver. So once this erythropoietin is produced, it specifically acts on hematopoietic stem cells in the bone marrow and causes the production of proerythroblasts. And this obviously it stimulates the process of erythropoiesis, and eventually mature RBCs are produced. So let's look at the mechanism by which erythropoietin increases the production of RBCs. So when there's hypoxia in the kidneys, this increases the levels of hypoxia-inducible factor 1, which is a transcription factor, and this also acts as a transcription factor for the erythropoietin gene. So this HIF1 binds to a region called hypoxia response element within the erythropoietin gene, and this induces the transcription of the mRNA, and eventually the mRNA would produce erythropoietin. And sometimes hypoxia in other parts of the body besides the kidney can also stimulate the production of erythropoietin in the kidney. Other factors also promote erythropoietin production, like hormones like norepinephrine, epinephrine, prostaglandins, thyroid hormones, testosterone. So if somebody asks you what is the most important factor that controls the production of RBCs, then the answer is tissue oxygenation. And as I mentioned previously, tissue oxygenation is decreased by these factors, and a decrease in tissue oxygenation promotes production of erythropoietin, which in turn stimulates the production of RBCs. So when a person is at a place in which there's a low partial pressure of oxygen, like at high altitudes, the erythropoietin formation begins very early, within minutes to hours, but no new RBCs appear in the blood until 5 days later. So why is there a gap of 5 days? This basically means that the function of erythropoietin is to act on the hematopoietic stem cells in the bone marrow. So once it acts on the hematopoietic stem cells, it causes the production of proerythroblasts, and it causes the different cell stages to pass rapidly, and this speeds up the production of RBCs. And so this process continues as long as the person remains in the atmosphere of low oxygen, or until enough RBCs have been produced to carry the amount of oxygen required. Another important thing to note is that if both kidneys fail, or if both kidneys are removed or destroyed by some disease, then the person would develop anemia, because the kidneys have a major role in producing erythropoietin. So they produce 90% of the erythropoietin. So obviously, in the absence of erythropoietin, there would be very little RBC production, and the person would develop anemia. Vitamin B9, which is folic acid, and vitamin B12, which is cyanocobalamin, are two important vitamins which are very important for the maturation of RBCs. This is because these two vitamins are required for the production of thymidine triphosphate, which is required for DNA synthesis. So when these two vitamins become deficient due to any reason, for example, due to some intestinal disease like sprue, decreased absorption of these vitamins, or a dietary deficiency of these vitamins, then the person develops maturation failure anemia. So the mechanism of this anemia is that when either folic acid or vitamin B12 are deficient, then DNA synthesis is reduced. And when DNA synthesis is reduced, the nucleus can't mature. So there's a failure of nuclear maturation and a failure of cell division. So if the cell can't divide, then it just starts to increase in size. And eventually these RBCs become really large, and they are called macrocytes. And these macrocytes have really they have fragile cell membranes and they easily rupture. So this is why this is called maturation failure anemia. So another type of maturation failure anemia is called pernicious anemia, and this is specifically caused by the deficiency of intrinsic factor. Intrinsic factor is required for the absorption of vitamin B12 because it binds tightly to vitamin B12 and protects it from being digested. So intrinsic factor can become deficient when there is atrophy of the gastric mucosa, or when there is basically a destruction of the gastric parietal cells that produce intrinsic factor, or when there's removal of the stomach, and other reasons as well. So any factor which causes the deficiency of intrinsic factor will cause the deficiency of vitamin B12, which will in turn lead to maturation failure anemia, and this specific type of maturation failure anemia is called pernicious anemia. Also, because significant amounts of vitamin B12 are stored in the liver, so it takes several years, 3 to 4 years of defective B12 absorption for the maturation failure anemia to develop.